Hi, my name is Daniel Sivalingam, and today I'd like to tell you a little bit about the work I did as a Bridges student in deriving distal lung epithelium from induced pluripotent stem cells. The laboratory that I worked in was a lung laboratory, and one of the diseases we studied was a disease called idiopathic pulmonary fibrosis. Idiopathic pulmonary fibrosis, or IPF, is a disease of the lungs that impairs the patient's ability to exchange gas. It affects mainly the elderly, with over 2/3 of patients being affected over the age of 60. As far as treatment options for IPF, there aren't any, aside from lung transplantation when gas exchange becomes so bad that it becomes a medical necessity. Some of the hallmarks of the disease are a dying off of lung epithelial cells that facilitate gas exchange in the lungs, an overproliferation of alpha smooth muscle expressing myofibroblasts, which form what are called fibrotic foci in the lungs, and a loss of normal lung architecture. So over here you can see a section of normal lung, and right here you can see the open areas that allow gas exchange to occur in the lung. Now in the IPF lung here, you see that the normal lung architecture has been crushed, and you see these areas of what are called fibrotic foci, which contain the myofibroblasts. So the question is, how is IPF studied? And currently it's studied using an animal model known as the bleomycin mouse. Bleomycin is a drug that was originally used for treating squamous cell carcinoma. However, one of the side effects was found to be pulmonary fibrosis. It was found that this effect is also seen in mice, and this model has been used for quite some time. One problem with this model is that, unlike IPF, which is permanent and progressive, the pulmonary fibrosis in the bleomycin mouse resolves after about 1 month on its own. Thus, a better model of IPF is needed. The goal of our project was to create a better model for IPF, and we wanted to do so using induced pluripotent stem cells. For those who don't know, induced pluripotent stem cells were discovered by Yamanaka and colleagues in 2007 when they took dermal fibroblasts, added 4 reprogramming factors, and were able to generate cells that were pluripotent and thus able to make tissue from all 3 germ layers. We wanted to use this important technology to study IPF. The overarching goal of our project was to create a better model of IPF by differentiating iPSCs into distal lung epithelium. The major roadblock in that was that no one had yet made a protocol for generating distal lung epithelium from stem cells. So the focus of my project, and what I'm going to talk about today, is how we generated a protocol for deriving these distal lung epithelial cells. Well, first, I'd like to talk a little about the iPSCs that we used. We teamed up with a lung transplant team at UCLA who were able to get us lung samples from IPF-affected lungs as well as healthy donor control lungs. The fibroblasts were isolated, which you can see here. We added 4 transcription factors, the same 4 that Yamanaka used, and reprogrammed the cells to stem cells after a period of 6 weeks. So after 6 weeks, we had cells that looked like this, that were consistent with the morphology of iPSCs, but we had to make sure that they were indeed iPSCs. In assessing the iPSCs, the first thing we looked at was the karyotype. We wanted to be sure that there were no gross genetic abnormalities in the cells, and we sent the cells out to Cell Line Genetics. They did a karyotype for us, and they all came back normal. The next thing we wanted to do was see if the cells expressed markers consistent with iPSCs. To do that, we used OCT4, SOX2, and NANOG. We found that the cells were positive for all 3 markers, consistent with iPSCs. Lastly, we took some of the cells, injected them into mice testes, and after a period of 6 weeks, these tumors known as teratomas formed. It's expected that in these teratomas we could find tissue from all 3 germ layers, showing that the cells were pluripotent. And when we sectioned them and stained them, we indeed found that. So here you can see neuroepithelium, which comes from the Endoderm germ layer. Here along the edge you can see pseudostratified columnar epithelium, which comes from endoderm, and at the bottom here you can see cartilage, which is comes from mesoderm. At this point, I'd like to note that the generation of the protocol was done with a donor iPSC line D3. After assessing the iPSCs, it was time to work on generating the protocol for differentiating cells into distal lung epithelium. Here you see a basic schematic that we're going to slowly fill in with what we use to recapitulate development and markers that we use to assess the cells at each point. The first step in our differentiation is a generation of definitive endoderm. Definitive endoderm is an early step that happens around gastrulation, and the lungs as well as many other organs arise from the definitive endoderm. The definitive endoderm is formed by nodal signaling in the developing embryo, and to mimic that we used activin A, which utilizes the same receptors, as well as a PI3 kinase, which has been shown to, in combination with activin A, generate more definitive endoderm. After 4 days, we assessed using flow cytometry. So let's take a look. Here at the top, we have two morphological pictures from day 1 and day 3. Of note, there was some cell death upon addition of PI3 kinase inhibitor, which has been reported in the literature. You can see changes in morphology with the cells taking on a larger, spikier appearance. When we looked at markers of definitive endoderm, we found that over 95% of the cells were positive for CXCR4, a very important marker in definitive endoderm, and that 76% of the cells were positive for CXCR4, c-KIT, and EpCAM, so all three markers. Once we'd made definitive endoderm, our next step was in making Foregut endoderm. So in the developing embryo, Foregut endoderm is driven by a few inhibition steps, the first of which being an inhibition of BMP, and the second of which being an inhibition of Wnt. During both steps, TGF-beta is inhibited. To mimic that, we used dorsomorphin for inhibiting BMP. We used SB431542 for inhibiting TGF-beta. We used IWP2 for inhibiting Wnt, and we continued using SB431542 for inhibiting TGF-beta. After the two inhibition steps, we saw an increase in both SOX2 and FOXA2 from day 5, with a lowering of them at day 11 and day 15, which is expected, as at that point we are ventralizing the cells. So once we'd anteriorized the cells into foregut endoderm, we then needed to work on ventralizing the cells into ventral foregut endoderm. Ventralization is a very complex process and utilizes multiple different factors. Here we recapitulate development using a small molecule agonist of Wnt called CHIR. We also use BMP4, FGF10, FGF7, and retinoic acid, all shown to play roles in ventralization of the lungs from the anterior foregut endoderm. So at day 9 and day 15, you can see that cells are taking on a much flatter appearance, consistent with epithelial cells. Eventually at day 15, forming a nice epithelial monolayer. At day 9, we looked for markers of ventral foregut endoderm, PAX9 and TBX1, and we found them to be positive. At day 10, we looked at a very important marker for ventral foregut endoderm, a transcription factor called NKX2.1, which we found by both qPCR as well as by IHC. We then looked at SOX9, which is also a marker of ventral foregut endoderm, and found it by qPCR and by immunofluorescence as well. Now, NKX2.1 is a very important marker of ventral foregut endoderm, but it's not just expressed in ventral foregut endoderm, it's also expressed in forebrain. And so to show that the cells that we were making were not forebrain, but were instead epithelium, we used two epithelial markers, E-cadherin and cytokeratin 5, showing that the cells were expressing both all three, NKX 2.1, E-cadherin and cytokeratin 5 showed us that we were indeed making ventral foregut endoderm. Once the cells have been ventralized, the next steps are a distalization of the lung epithelium, which is where the lungs branch outwards, and then a maturation into type 1 and type 2 alveolar cells, which you can see here. So we're going to talk a little bit about that. To distalize the cells, we used some of the same factors we used in the ventralization step, namely FGF 10 and 7, as well as the Wnt agonist CHIR. What we did change, though, is we dropped retinoic acid as well as BMP from the mix, as both were shown to lead cells to a proximal fate. After treatment with that, we then worked on maturation. We used a cocktail consisting of a steroid called dexamethasone, cyclic AMP, as well as isobutylmethylxanthine. This cocktail has been shown in both fetal and human lung explants to rapidly mature distal lung epithelium. To assess, we looked at a few different markers. We looked at surfactants protein B and surfactant protein C, which are both markers of type 2 cells, which you can see here. We got widespread expression of surfactant protein B with somewhat lesser expression of surfactant protein C. We then looked also at aquaporin 5, which you can see here, which is a marker of type 1 alveolar cells. We saw a much patchier expression of aquaporin 5, seeing that we had expression of the surfactant proteins, we thought that perhaps we might be getting lamellar bodies in the cells, which are structures that transport surfactant protein to the cell membrane to be exocytosed. So to look for that, we used a stain called Nile red. So here you can see a good example of Nile red staining that we got from the paper that we also got the protocol for using Nile red from. And over here you can see Nile red staining from the alveolar epithelium that we generated. Here you can see different intracellular lipid droplets, which are consistent with what you'd see in alveolar type 2 cells. So this is the final protocol that we have generated, and so let's move on to some of the conclusions. In conclusion, we have generated iPSC lines from IPF-affected patients as well as normal donor lungs. We've also generated a protocol for deriving distal lung epithelium, specifically surfactant protein B and C expressing alveolar type 2 cells and aquaporin 5 expressing alveolar type 1 cells, and we did this utilizing a stepwise progression which recapitulated lung development in vivo. Doing so, we've developed a useful tool for studying IPF as well as other diseases of the distal lung, and that can be possibly used in potential cell therapies. With this, I'd like to thank you for listening and thank Bridget Gompertz and her lab for taking me on for this work.